We are joined tonight. This webinar is being hosted by the NorthStar DMD network, alongside Muscular Dystrophy UK, Action Duchenne, and Duchenne UK. We have representatives from each of these organisations on the panel tonight. From Muscular Dystrophy UK, we've got Kate and Rob. From Action Duchenne, we have Sophia, and from Duchenne UK, we have Alex and Alessandra. For all the attendees, you'll see at the bottom of your screen, you have a little tab that says Q&A. If you'd like to ask any questions to the panel as we go along, you can just put them in that Q&A box and we will answer them either as we go along or at the end. So I'd like to introduce Professor Francesco Muntoni, who's going to talk to us tonight about, give us a little background to the EMBARK gene therapy study and talk to us about the results. So I'm going to share some slides on screen now, and then Professor Muntoni can take us through the study. Thank you.
Thank you.
Oh, we have a bit of distortion there. So just a second. Can you try speaking again, Professor Muntoni?
I think that there is a problem because I am in two. Let me just rejoin because I'm going two places and just one second.
No problem at all.
Can you hear me now?
Yes.
So anyway, that's fine. I'm more used with Teams, so I apologize. I was the one who was creating the echo, and I was thinking, who is creating this echo? I wanted to blame somebody else. It was my fault. Anyway, well, thank you for this. As you realize, this is a short notice, both in terms of for us to get together. I suppose more importantly, for us to understand what this data mean and have sufficient data. But I think we need to start discussing what we have. We didn't have enough time to meet with the other colleagues from the Duchenne Clinical Network. So this is the first time we meet, and hopefully some of the colleagues will also be present. So we will have a combination of families, advocacy group, and also colleagues. And I have just 5 or 6 slides just to get us up to speed, because I don't think this is a place to present hundreds of slides, but is more to tell what we have. And if you can move to the next slide. Yeah, so this is the only, in inverted comma, technical slide I have. And this is the most important aspect, is that this is an AAV vector that has been used largely by Sarepta. It is slightly different from AAV vector that I use by others. The shorter gene, the microdystrophin, is not fundamentally different, although it's not the same compared to some of the other gene therapy in clinical trial, for example, the one from Pfizer, the one from Genethon. In fact, it's very similar to the one from Genethon. It's not the same as the one from Solid, but I think these changes are relatively minor. And I think the promoter is what drives the production of the protein in muscle and heart. Next slide. And you can stop me at any time if something is not clear. But I think, again, I know that 80% or more of the people here know much more about gene therapy than what this few slides showed. But the EMBARK study is a 2-year clinical trial. The study is divided in 2 parts and recruits children between the age of 4 and 7. As you know, just want to go back.
Oh, excuse me.
No, it's okay.
Oh, sorry.
Yeah. So the study has 2 parts. The study is the first year. Children will receive either the gene therapy or a placebo, and then after the first year, everybody gets the other part. The study is double-blind up to this point after 1 year, But of course, after the first year, everybody will have received the AAV gene therapy, so it does not become a placebo trial anymore after the first year. So at this point in time, every child has received the gene therapy. However, we only know about the result of the children who have been recruited in the first year of the clinical trial, and therefore some receive the placebo, some receive the active drug. Next slide. So I think these were the inclusion criteria. The children had to be between 4 and 7. There are some specific mutations that were excluded, but we're not going to go there now. I think that importantly, the NorthStar score had to be between 16 and 29. So the two extremes were excluded. And the time to rise must be less than 5 seconds at recruitment. So to some extent, these were, broadly speaking, reasonably high functioning people with Duchenne. Next slide. So what were the primary outcome? The primary outcome, every time there is a trial, there is the primary outcome. That is what you say, I think this will be what we plan to achieve. Then there are also secondary outcomes. The primary outcome was the score with the NorthStar, and the ambition was to identify difference between those children in placebo and receiving the AAV gene therapy after 1 year that was statistically significant and clinically meaningful. Next part. The secondary outcome measure were some additional information regarding the items in the NorthStar, because there are particular techniques to calculate that. But also there was the 10-meter walk, the time to rise from the floor. And for the first time in a clinical trial, the stride velocity, that is a methodology that uses a very sophisticated Apple Watch to tell what is the level of activity, but also the speed of this activity for a period of 20 days at a time in the community. And finally, the protein expression. Many children, not all, but many children had a muscle biopsy. Next part. So the results, you're aware of them, but I think that the important point is in bold that while there was an improvement in the NorthStar in the treated children, this did not differ in a significant way from the placebo. As you can see, also the placebo patient improved, and therefore the difference was less than a point in the NorthStar and was not statistically significant. And this is important because this was the primary endpoint, and therefore from that perspective, the study did not achieve the primary endpoint. Next part. The secondary endpoint were, if you like, at least for the study, a little more rewarding, because the functional secondary endpoint, such as the time to rise from the floor, the 10-meter walk, the stride velocity, that is this Apple Watch, they were both statistically significant and also clinically meaningful, meaning is not just, that there is a minimal, just about significant result there, but this is unlikely to be of any importance. There are techniques that have been used to understand whether the changes may be clinically meaningful, and these changes were clinically meaningful. A result that was not statistically significant, but also trend, a bit like the NorthStar in the right direction, was the time to do 4 steps. Next part. I don't think there was any new information, meaning additional problem related to the safety. Next part. And I think it's important to recognize that the data we have are high-level data. We still miss a number of things. For example, we miss the information regarding the points gained and lost in the NorthStar. I'm not aware of the detailed dystrophin level being presented. There is other scores that, for example, the upper limb extremity score, and patient-reported outcome measure that at the moment, presumably they have been analyzed, and therefore they're not available to any of us. Next part. I think that where from here, well, the study is continuing, of course. This is the first year of a 2-year clinical trial. I think that what will be important is to continue clearly to make sure that children are doing well from the safety and assess the efficacy. I think that one probably on one end undesirable issue is the fact, undesirable especially from a regulatory perspective, that now the placebo integrity of the study has been broken because all patients have received the drug. So external comparator will be necessary if the sponsor wants to demonstrate that or show that there may be efficacy. I think now we'll have to anchor the data of the treated patient to natural history data. Next part. And I think this just summarizes what I said. There will be more data that become available, hopefully soon. I think that is not a surprise that from the press release from Sarepta that they will be entering discussion with their regulator, that is FDA, regarding the possibility to extend the current label that is only for the 4 to 5 year old. But in Europe, this will be discussion that Roche will need to start making, but at the same time, I think the question will also be, and I'm sure Roche will be interested at some point to hear what the medical community in the UK feel about this, because I think somebody will have to knock on the door of MHRA. This will have to be Roche to know whether there may be the possibility to consider access to the, access for filing as things stand today with the failed primary endpoint, but with secondary endpoints that are going in the right direction, or whether to wait until the completion of the second year of the clinical trial or any other consideration that the Roche and Sarepta may have. I think that, as you expect, the result, I expect as an investigator in the clinical trial, I expect to have much more data to share, but also for me to understand, because the results are not exactly what had been expected. There is some area of positivity and other of negativity that I hope I am presented in a way that is unbiased. But I think that this is now time for the discussion. People will have questions, and I don't think I have any more slides, but Matthew, just go forward to see whether that was the last.
That's
Fantastic.
That's correct. So I will stop sharing there.
Yeah.
So yes, we've received a couple of questions via the Q&A, which I'll just put to you now. So the first of these is kind of something you just touched on a second ago, asking kind of overall, do you feel these results are positive, and are we getting closer to a gene therapy being approved in the EU and in the UK?
A very simple question.
Mm.
Well, I think that I think again, I think it is important to understand two things. Firstly, the results are not exactly what the company had expected, because otherwise they would have said, we'll measure another thing and not the NorthStar. I think if you let me answer in a different way, that I think is the way I see it. So for example, you probably are aware the stride velocity has just been approved by EMA just last few weeks as a primary. So had the stride velocity been put as a primary, then we will all say, well, the drug has worked, and therefore, where is the problem? I think there is a problem, and the problem means that we need to understand more, because the question is the change substantial to really EMBARK in such an important thing or not. And I think that is a matter that I don't think is completely black and white. I think we'll be dishonest to say, I think the drug works, because I think it will be very difficult to recapitulate all the secondary outcomes that go in that direction and the NorthStar that moves in the right direction. However, I think it will be better to address that question once we know exactly. I'm very curious. I would like to discuss and see the result of the patient we treated. What was the NorthStar at recruitment? We have only aggregate data. Was there protein in this patient? It would be quite different if Sarepta tells me or tell us, well, the average level, I'm using example now, is 5%. Well, that's not particularly impressive. But if they say, well, it's 60% as an example, that's quite different. And although we know that, we have not yet enough information on what will be the role of this surrogate microdystrophin. If there was a very small fraction, that will not be a good news, I think. But I think it will be very different. So I think we are lacking information to be able to honestly answer that question. And my sense is that I think we need a little more time to date. I think that, is not a completely failed trial where there is, we have been involved, unfortunately, in trial where the placebo and the treated patient were absolutely flat. You could not distinguish one from the other. This is not the case. However, in all fairness, the amount of treatment effect at 1 year appeared to be less than had been expected. And also, the gene therapy, you can only do one. You cannot, you stop another drug and then you start with another clinical trial the day after, or after 3 months usually. With gene therapy, you cannot do it. So I think I suspect it's premature to give an honest answer to that question. But especially if there are other clinical colleagues and anybody who wants to, I'm telling you what I'm not a Sarepta representative or a Roche representative. So what I mean is, I'm telling you what I think. That other people may have very different views, and is what we're meeting to discuss different views, not because I'm telling you the truth. I'm not that kind of person who has the truth.
Great, thank you. We've had a couple more questions. I'll just try and summarize. A couple of them have asked quite a similar question, which I'll just try and paraphrase here. Asking about the placebo group and why that might have improved by 1.9 points over the course of the 52 weeks. Is the suggestion that this is a result of steroids, or would the results perhaps have been more favorable had the patients not, the placebo group, not improved to that extent?
Well, I think, again, this is a simple question, but it's such a crucial question. And to some extent, unless the people at Sarepta are completely unprofessional, this level of improvement in the placebo took them by surprise, I suspect. And I think this means multiple things. Firstly, that unfortunately, this is maybe what people don't want to hear, but unfortunately, we learn something new every time we do a clinical trial. And I think it would be nice if it wasn't like this, but that's the truth. So there were a few things that we didn't know. How much they play a role here, again, I'm not telling the Roche or Sarepta truth. I simply don't know, and presumably they don't know either. So the patient I think that the improvement of 1.9 point, I think, in this age range is not typical. And a possibility is that because of the placebo nature of the clinical trial, all patients, even those not receiving the gene therapy, have a bump of a much higher dose of steroid for a number of months. There is no question, and this is data that you can dig from the presented data from Sarepta in the past, from their patient in the placebo, that there is a bump that lasts a minimum of 3 months, where this patient clearly diverge from their previous trajectory. I don't think we have enough information. What is the medium term, say at 1 year, effect of having bumped the steroid for 3 months at much higher doses than the one we use in standard of care. So this may have affected. I'm not saying it has, it needs to be clear, but this may be an area where there was perhaps lack of sufficient planning or information regarding what would have happened. And again, I would be grateful if any colleagues or anybody has other position or other things. I'm telling you what my eye sees, but I don't have the truth.
That's great. That's of course why we're here. The next question is kind of a question about the study results. Was the level of dystrophin measured in the patients at this stage?
It was.
And do we have that data available?
I do not have that data. I do not have the data, but I expect this type of data will become available soon, because it's in the interest of the sponsor to make this data available. I think in actual fact, as publicly traded company, they cannot hold the data; it becomes illegal. So they will have to release. But and I've been involved in many clinical trials, and understandably, the company will not like to say there is 19%, and then the day after they say, well, actually, it was not completely right. It's 18.3%, because this is a big problem for them. So there are a number of quality control before the data is released. But I would be surprised if this is not released in the next week or 2 weeks. I don't think this will be released in 3 months' time. This must be coming very, very soon.
Excellent. And kind of related to that, do you have any views on why you think the primary outcome wasn't achieved? Does it suggest that the gene therapy wasn't working in the sense of producing dystrophin, or could there be some kind of confounding factors?
Well, I think again, the dystrophin production will have to wait. I think that there may be multiple reasons. I suppose one is that in actual fact, let me summarize a few. One is there may not be enough dystrophin. The second is that the microdystrophin is not as good as people would like it to be. The third possibility is that when I give my lecture to medical students, I try to explain, and I do the same to companies, but sometimes they listen, sometimes they don't, is what is the dystrophin does? What is the effect of dystrophin? Now, Matthew, we only have known each other for a few weeks. I do not know if you run, but if you run and you want to run faster or longer, I can guarantee that dystrophin is not the protein you need to take to become stronger or run faster, because that's not what dystrophin does biologically. So dystrophin is not a muscle enhancing drug. So dystrophin is a drug that really protects muscle from damage. So I think you do need, therefore, so I think it's probably unrealistic to expect a big divergence, a big improvement in the short, medium term compared to the untreated patient, because that's not what dystrophin does. And I think that therefore, I think there was probably there is a misconception regarding what to expect from having dystrophin restored. And therefore, the question would be, is a 1 year timeline sufficient to have divergence, especially at the time the placebo are also improving? I have no idea. I do not know if which one of these is true, whether each of them is partly true and they all contributed to this. I think that I suppose, as I indicated at the very beginning, while it's important to remark that primary outcome has failed, there is no other way of saying this. I think that the fact that all the other secondary outcome had been moving in the right direction, actually 3 being statistically significant, that probably suggests that there is a drug effect. But again, we will all need to understand whether the drug effect is sufficient for what we as physicians wanted and what the family wanted. I do not know that yet.
Okay, thank you. We have a question from a parent here, which I'll just ask straight up. If I, the parent, was able to get this therapy for my child, would you recommend I do this or wait for more results?
I would like this to be discussed, not in a forum. I think I would like to discuss with every single individual families with what are the data that are available, understand better the individual priorities. You know, in this call I have now 6 or 9 people. I can guarantee we don't all eat the same thing. We don't all drink the same. You know, we all take different style. I cycle to work, and some people saying to my mom, "I'm stupid because the risks are too high." So we are all different. The point is that the risk benefit or the perception are different. I don't think we'd be wrong either of those two choices, quite honestly. But I think that there may be practicalities of that need to be discussed. The age of the child may need to be understood and discussed. You know, there are multiple aspects that is a bit difficult to give a blunt answer, yes or no.
And ask one more quick one. With a view to approval of the therapy in future, is it theoretically possible for the relevant authorities to approve the therapy, even though the primary endpoint wasn't met?
I think that in theory the answer is no. Certainly for a common disease, the answer will definitely. If this was a drug for diabetes, I think this would be a problem. Meaning, if the company was really committed and thought the drug was working, we'd do a different study. We'll have to do a completely different, start from scratch, and take a maybe slightly different population, blah, 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 what we've been discussing. For rare diseases, I think that there is a range of negotiation. And I think if you go and look at I think the interaction that the companies already, I'm referring what I've read myself. If you Google what's happening, you get updated. That's all the information I have. But I think that the company is in discussion with FDA because of the coherence of the secondary endpoint. And I think that there will be a discussion that they need to have. For rare disorder is not impossible, but clearly is a hurdle that I'm sure Sarepta didn't want to have.
Thank you. We do have a couple more questions, but I'm conscious that we have a few more people on the panel. So I'd just like to kind of open it up to the rest of the panel to ask whether anyone on the panel had any questions or any comments on what we've discussed so far tonight. If not, I'm happy to kind of continue the discussion with the submitted questions.
I'll ask Francesco a question. I'm asking this as a mum, Francesco. So one of the things that I sometimes am concerned about is the primary endpoint of the NorthStar, and wonder, is it sensitive enough to see the changes that are happening? And I only ask this because I think about my own son, and he did start not this treatment, another treatment, and me and my husband both felt we saw an improvement in him, but his NorthStar didn't change. But myself and the physiotherapist both noticed, for instance, his jump was higher and how he performed the jump was better. So, for instance, he'd squat into the jump and he had more height. But because he'd always been able to clear 2 feet off the floor, he still scored the same 2 points, even though we could visibly see an improvement. And I do sometimes wonder, over these shorter time points of a year, are we going to see these small changes? I agree with you. I'm looking forward to hopefully seeing the 2-year data. And I do I'm encouraged by the stride velocity data because I think it's a little bit more sensitive in detecting those smaller movements. So I just wondered what your thoughts were on that.
No, I again, as a disclosure, we, our group and together with all the UK, where we were involved in the NorthStar. So I have a little of an emotional attachment. I'm saying now so that it is clear that whatever I'm going to say, I'm conscious of this. So I tell you what I think, that not what my heart if you like, not because of my emotional attachment. I really don't care about if you like, the fact where we have been involved. I think you're right. You're right but I think that is also why it would be very interesting to see the scores of the children. How many how many were on a 2? Because you are right that if you are in a 2 and you do things better, then you would miss that in the NorthStar. We were not totally unaware of this, and that is why the 10-meter walk and the rise from the floor are part of the NorthStar, because we had considered at the time, because this was at the time when corticosteroids were introduced, that if you are ceiling in the NorthStar, what you do. And the 10-meter walk and the rise from the floor that were positive here are, if you like, were meant. I'm not saying is the perfect, I'm not trying to justify that the NorthStar is perfect the way it is, but I think that was the first attempt to say we may hit a ceiling and describe, and what you describe is also what we see in clinic, especially now that we are hitting the ceiling. You know, the NorthStar was established at the time there were no steroids. Children essentially could never, very rarely run. Jumping was exceptional. Now that is becoming much more common, and therefore I think what you are suggesting is right. And I think that the digging into the shift of the how many people were already on a 2, but also which is there any change that may give a little more granular information. But I think you are right that the stride velocity or the 10-meter walk and so on may collect these changes better. I suppose I always say there is never a free lunch. There is also still a problem, because while for the NorthStar there has been a lot of work to determine what is the minimal clinical important difference, for the 10-meter walk, the stride velocity, the rise from the floor, this is only be done via statistical construct. So because it's very difficult to ask me and say, if I walk 0.5 seconds faster, 10 meter, that is clinically significant. For the NorthStar, that work has been easier. So from that perspective, I think that I think is probably the technical discussion regarding the correlation between minimal clinical important difference of all these different outcome measures. But to me, what you say makes sense, and to some extent is why I think we need not to be completely despondent for the lack of the primary in the NorthStar, because exactly the reason you said, and I would not say this if 10-meter walk, rise from the floor, and the stride velocity were flat. If they were flat, I'm sorry, they were flat, but they were not flat.
Right, thank you. If there's nothing more from the panel, I'll get in another quick couple of questions from the chat, just so we can answer as many as we can. There's a question here about the side effects that were encountered during the trial. I'm not sure if you're aware of them. Are you aware of what the kind of common and less common side effects were?
I think that, firstly, I'm a principal investigator in the trial. Secondly, I described for Sarepta some of the side effects that we published in the New England Journal of Medicine a few weeks ago. So yes, I'm aware of what the side effects are. I think that there have been a few significant adverse events that are one in particular, two myositis. One was clearly transgene induced. The second one is less clear what it was. It would be fair to say, however, that and a number of patients had developed a liver problem that is almost common in AAV gene therapy. I think it would be fair to say that the profile from the early data from the what has been presented already from previous open label study really has not changed in this age range. I think that all AAV gene therapy at the same time, age and weight may be important. So it's important to realize this is in the age from 4 to 7, would be fair to say, has been on the whole reasonably well tolerated. Has the severity of adverse event has been limited in the number of patients with severe adverse event. If you go to other IV gene therapy trial or commercial gene therapy for spinal muscular atrophy, you will find a profile that is not fundamentally different and certainly is not better.
Great, thank you very much. Well, we're coming up towards the end of our time now. So I was wondering whether you, Professor Muntoni, or anyone else on the panel had any kind of final comments, any kind of summary of the of what we've discussed this evening before we draw this to a close.
Well, I think that I would like to have more data, and I would like to understand the level of dystrophin, and I would like to understand a little better the following what also Alex mentioned, whether our possible interpretation is true, and whether the number suggests that some of these patients were at the top. And I think will be probably discussion with the medical community and with the families of addressing the question that one of the family asked, whether if the drug was available today, would we give it today? Would we wait another year? This, I think, I would not be able to answer honestly this question today, because I do not have a lot of information. But at some point, hopefully in the coming weeks, this will become available. And then maybe when that is available, we could, if colleagues feel and the families feel this is not a waste of time, we briefly reconnect to see where we are. And I think to see whether this is, whether the additional data change what we are saying, whether we are equally confused, we are less confused in one direction or the other. It's a bit difficult to discuss about things without having all the data.
Great, thank you very much. I will add, there were a couple of questions we haven't had a chance to cover. So we kind of in the NorthStar team will be able to kind of discuss those questions away from this venue, and we and the advocacy organisations that are here can publish a Q&A after this event's taken place. If there's anything specific that we haven't talked about tonight that you'd like to ask about, you can either get in touch with myself or with any of the advocacy organisations who are represented here. So if nobody else has anything you'd like to mention now, then
I will only like to thank you for arranging this, because I have no idea how to do one of these things, as it was clear with the echo at the beginning, but also the representative of all the advocacy group, because I think this is, is much easier if we're all together here. This is one of the good things about our country is, on the whole, whenever it's possible, we try not to say, well, we do this in this way in London, but not in Swindon. And then, we try as much as we can to be aligned. And I think it's nice to see that we are aligned.
Excellent. Well, on that note, I'd like to thank everybody that's attended the panel tonight and everybody that's attended as an attendee to the webinar. As I say, if you have any further questions, please do get in touch. And otherwise, thank you very much and good evening.
Thank you very much. Good night.